The aim of this practical is to perform an ACTN3 R577X genotyping test. It is taught as part of the MSc in Molecular Exercise Physiology at the University of Aberdeen. Like every PCR reaction, we have to set up a master mix. We need molecule water, buffer, DNTPs, the ACTN3 primers, magnesium chloride and TAC. We have to melt our ingredients thoroughly on ice. And then in a special fume hood, add everything together. This will be the master mix. The last bit is a TAC polymerase. It is an enzyme, and we only add it at the end in order to have it out of the freezer for a very short period. Here it is added and it goes immediately back into the freezer. The master mix is mixed and then it is pipetted into small tubes or wells of a plate. Here you can see the green master mix in some wells. After that we remove the master mix in the wells from the fume hood and add the genomic DNA. We separate the two steps in order to prevent genomic DNA contamination. Two DNA samples have been added here. Also we add some water as a negative control, because if we add water rather than genomic DNA, nothing should be amplified. We seal the wells, vortex and spin it down. The last two steps are optional. Here's a PCR reaction. It consists of a melting step where the DNA strands separate, followed by an annealing step where the ACTN3 primers bind to the specific fragment of the ACTN3 gene, and then a step where TAC polymerase extends the DNA product. This is a display of the thermal cycler where we have just added our reaction and the first cycle has been completed, and now the temperature goes back up to 94 degrees for another melting step. The DNA fragments separate. After that, the primers will anneal to the specific position on the ACTN3 gene, and then the extension follows where TAC polymerase binds to the primers and extends to form another double strand of the PCR product. We perform 30 to 35 cycles of this reaction. Once the reaction has been completed, we could already visualize the DNA. However, in this assay, we have now to digest the DNA, and we use an enzyme called DDE1. It cuts DNA in specific sites. Now, for the ACTN3 genotype, in position 577, the DNA can either encode an arginine or R, or alternatively a premature stop codon or an X. If our product contains the DNA that encodes an R, it will be cut once. If it contains the DNA sequence encoding a stop codon or X, it will cut the product twice. Again, we add our product to a thermal cycler and the enzyme will work at 37 degrees for 12 hours. And after that we denature our enzyme at 65 degrees. Here the reaction is added to the thermal cycler. The last step is to visualize our DNA products. We add agarose to a conical flask and then TBE buffer. It is mixed together and added to a microwave. We now carefully have to microwave until the solution is clear. Now we have to cool it down so that there is no fume evaporating. We now have to add ethidium bromide, which is highly toxic, and blue nitrile gloves have to be worn for this step, but also for most other steps. Here we add the ethidium bromide to the slightly cooled down solution. It is thoroughly mixed. And added to our gel casting stand. Combs are added, which will provide the wells. Once the gel is solidified, we remove the combs and add TBE buffer. We now add our samples. In the first lane, we usually add a marker, which is a DNA ladder comprising of different lengths DNA fragments. It is an important standard. Here we add our product, which has been digested by DDE1. The gel setup is assembled, plugged into the power pack, and we let the gel run until the DNA has separated sufficiently. Once that has finished, we add our gel to a UV light imaging system, and here we see the results. The bottom band is our primer dimers, but above we see different band patterns indicating whether someone is homozygous or heterozygous for the R, X or RX alleles. We then have to remove the ethidium bromide gel. If you're interested in the MSc, please email Henning Wackerhage.